water sense, including hydrogen cycles. You're going to hear about monitoring assessment plan. This is the acronym MAP. That's been thrown around a lot. And I think you should know that if you look at the literature at restoration plans, you'll find that the MAP, as part of CERP, is somewhat unusual. And it's unusual in that it's sort of hypothesis-based. What's very common in the literature, we monitor sort of upper trophic level organisms, maybe we'll monitor the process or the driver. That's about it. But CERP and MAP are basically sliced from top to bottom. They did a hypothesis sort of structure. They arranged their attributes or their performance measures kind of along that line. So what I'm going to do today is I'm going to talk a little bit about the trophic cluster of CERP, which is really the water driver, sort of the aquatic fauna in the middle, and then our top predator in this case is wading birds. But in terms of CERP in general, it would also be alligators. So that's the top level group. I'm going to talk a little bit about kind of a summary, kind of run through a number of studies that actually feed into this. One of the other things that's I don't think unusual about the map in South Florida is that there's lots of other work going on. It's not just this monitoring is done in isolation. Researchers are bringing in other studies to connect to it. And what that does for you is it gives you a little bit of extra ability to interpret the monitoring findings. So without further ado, I'd like to acknowledge the honors of the collaborators. Slides here. The trophic cluster of CERP could really be sort of grossly characterized as get the water right, get the hydrology right, you'll have restore those processes that produce mid-trophic level animals, the aquatic fauna that Sean was talking about, and then eventually you'll get the upper trophic level animals to follow. If you sort of put a little finer touch to that hypothesis and fill in a little bit, you get something we think that looks like this, where you have sort of local scale drivers, regional, and then other local scale. But basically, you've got factors that affect the fish populations, so things like nutrients, times this dry down. That gives you your population size. And then you have things that actually, that's the center area there. You have stuff that actually translates the population into these small patches that the animals can use. And that's factors like microtopography, water depth, water recession rate, things like that. And Sean, listening to the previous talk, mentioned some of those. So it takes the prey population, sort of reassorts it, and puts it in small patches that birds can use. So sort of that simplistic idea that when you get the water right, you have more fish and you have more birds, doesn't really hold up that straightforward. You have to sort of think about some of these other things as well. All right, so the early stuff that was on the Everglades really focused on the drivers, the water, monitor birds, and said, look, there's this relationship. It's almost certainly driven by food. And so that sort of started out our understanding of how birds respond. But it really missed for some of the middle parts, which are so important. Starting in the 70s, about 1973, Jim Cushman and other folks started monitoring the aquatic fauna in the middle of the system, long-term sites. They started doing habitat selection studies and started to fill in this knowledge a little bit better. And then more recently with work at FIU, they've looked at factors that affect or factors that produce these large fish populations. So what are the big drivers there? And they told us a lot about the fish populations, but now we still have to make that connection with the birds yet, right? And I argue when the map was being developed that if that's all you did, I was certainly going to fail. Because I looked at a lot of data, and there's no relationship between fish population size and wading bird nesting. And I'll say that again because there's $10 billion hanging on that relationship being there, and yet it doesn't exist. And the reason it doesn't exist is because you're missing that center part, and that's the availability. And that's the part that MAP sort of brought in and sort of filled in that last piece. So that's the picture. It's important to sort of understand that difference. You have drivers that produce the populations, and then you have translators that put those fish populations into small patches that the birds can use. And you need to be tracking both of those. My lab then brought in some other studies funded by other agencies that sort of brought in some of that ability to interpret those patterns a little bit better. The starting point for our work was work I did in the 1990s, and basically a set of experiments and said you can arrange wading birds along a bit of a gradient. It's a foraging gradient. I like to call it the cream skimmer crumb picker gradient. And you have animals on the left, like the wood stork, it's probably the most sensitive. It is clearly a cream skimmer. It finds patches of really high availability food, feeds down a little bit, and then when it gets to a certain level, the animals leave, and they fly away. And other birds, like gray egret, might just be arriving for the first time in that patch, and they'll stay in that patch, for days until the very last fish is gone. And you can go in behind these guys and good luck trying to find something that's left alive. Whereas storks will have abandoned long since left. So this turns out to be kind of a handy way to think about the bird community out there. And actually, you can go further with this and look at population trends, and what we find is the guys on this side are doing much better in terms of populations of nesting birds than those on the left. So there's a nice correlation there. All right, my colleague Joel Trexler and other folks have done a nice job of sort of starting that foundation between the drivers, the hydrology, and the fish populations. We've got lots of data like this out there now in the literature. These are for individual species, and you notice that's hydro period, or basins last flooding on the bottom, basically hydro period. And you notice that populations peak at different hydro periods. This is kind of important because it told us sort of what we need to get those large populations. But it still didn't have anything to do with the translators and how you take that and put it into bird food patches. And this is basically just a schematic sort of outlining that idea that you can have lots of fish in one patch, but the fish might not be very available to the birds. And in fact, if you measure the intake rates from the birds, they might do better on the patch on the left. So that's kind of the dynamic piece for me to keep in mind. This study is called the Fauna Concentration Project. This divides the landscape up into these units. You randomly sample or randomly put your sites out there. You pretty much have to get around by helicopter because it's done right as the marsh is drying. That's when the birds are first showing back. And so, this work I'm going to talk about here. And you notice some of it's down big cypress and that kind of herbaceous part of big cypress. But don't forget about them. It's not all cypress trees or plants. They're all open areas. Just a schematic of our sites. These are the species. So this is 2006 to 2010, 57,000. It's a moderate sample size. This cast of characters right here is pretty much, again and again and again the species you see in terms of plants. You can reshuffle them around a little bit from year to year, but that's pretty much it. Rat shrimps, fish, tillies, whitefish, bluefin gillies, crayfish. The interesting thing is, from a bird's perspective, really only the bottom three are really big prey items. So most of the animals in the Everglades don't show up that much in the diets of birds. So we're really kind of focusing on those bottom ones. We do modeling, and we've got 7 years in these, I think. The thing that pops out again and again is the recession rate, the microtopography, the wet season fish population size. So that there is the link, and there it is, by the way. There's your $10 billion connection now. We do have the wet, it had to be there. But we've got models now that show, yes, you need to have good standing stocks, good populations, but then you also need to have high microtopography, high variation microtopography. You need to have high recession rates or moderate recession rates, depending on your definition. And then there's some link with vegetation as well. The interaction with biomass and recession is basically telling us that it doesn't matter how much biomass of fish you have, how big your fish population is, if you don't have the right recession rate, you don't have way to work. And you can also flip that around. The interaction also tells you it doesn't matter what your recession rate, if you don't have that good standing stock, you don't have bird food. So that was kind of nice. I mean, some people say, well, geez, that's sort of illustrating the obvious, but the fact is now we have some quantitative numbers to put on that. This is just some data showing the patterns of those variables I just listed up there. Crayfish, there was kind of an interesting difference between fish and crayfish. The crayfish models were much more complex and they were less definitive, so lots more stuff affected crayfish. But the one thing that didn't affect them very much was recession rate. And I think Sean alluded to this, they don't really concentrate. They're not affected, they just go down into the mud. They're not going to be really high densities as a function of recession rate. So that was a little bit interesting. Vegetation had more of an effect on crayfish, so that tended to be positive. But from a bird's perspective, producing a lot of food is not the whole story. So we follow lots of birds around, measure where they landed, and if you look at the bird find perspective on this, you find that even if you define wading bird habitat in the way that you think represents what was good habitat, the birds still shift. This is the distribution showing random sites with the gray bars and the bird sites in the black, they still shift a little bit towards sites with a little bit less flock and a little bit more emergent vegetation. So there are even some subtleties there that we didn't appreciate early on. So we did some experiments digging at this a little bit deeper and said, well, what is the effect of vegetation density? And we found, here's our treatments. We had these little enclosures. I'll go back, by the way. If there are any good bird watchers in the crowd, if anybody can identify that species in the lower left there, you might recognize it. It's a South Florida native. It's a pink plastic flamingo with white. And you'll notice there's two at each enclosure. This is a really good trick, by the way, because the birds, when they're flying over, you want them to see your treatments. If you put two white decoys at each of your treatments, the birds will find them, and they'll be aware of them. So we use this now pretty much all of our experiments. It doesn't bias them, we put the same number out there, but it does attract them. And so you'll notice there's a group there in the center, and they're over on the far enclosure. Anyway, we use these quite a bit now. The two treatments we have were water depth, and then we stocked them different densities of vegetation. So there's the combinations for density. No submerged aquatics, moderate high, and then two depths. And if you look, there's the proportion that the birds should, if they're selecting these randomly, that's what you'd expect. But obviously, in this case, the birds selected all the shallow treatments, none of the deep. And they selected the moderate vegetation more than they did the other two types. Now, we were controlling for prey density here. So I think what happened was we basically, let me show you the next slide. This is actually foraging data, so intake rates. So how well did they do once they were there? And what we find is the only thing that really affected them was water depth. So the vegetation density didn't really have a big effect on them. So why were they going to just one, or preferring that one vegetation density? When we go back to our monitoring data, we find that the fish tended to be more dense in those sort of moderate vegetation density patches. So we think the birds were essentially anticipating a higher payoff, although they didn't get it because we controlled for fish, and therefore they didn't really see a bigger difference in the effect. Anyway, the point here is there are these subtleties that the birds are using, and we think it does have to do with their trying to the availability of fish, is queuing in on that. All right, so another study that relates to this was we went out and captured 201 radio birds and white ibises. We put radios on and we followed them around. I think one of the neat things about this study, probably the most sort of important thing for us and changed our thinking a little bit, was looking at recession rate. So there's Greg for great egret, white ibis with in the middle. And there's the availability in 2 years of recession rate. So availability and how much of the landscape had these different recession rates. And you notice in the 2 years, 2006 and 2007, the recession rates were nearly identical. 2007 was a much drier year ago. So the water level started out a lot lower, same recession rate. But the birds selected, that's the gray bar, they selected recession rates that were much higher in 2007. Both species did than they did in 2006. So we're like, well, what's going on here? What was different about 2007? So when we looked at our fish standing stock data and our availability data, what was different was 2007 was very low. So starting in the dry season in 2007, there wasn't a lot of food out in the landscape. And so birds were selecting sites with higher recession rates. They were using recession rate to compensate essentially for low standing stocks. And this was kind of important to us because now it's like, all right, there isn't a static recession rate. And as we talk here, there's a meeting in the water management district where the Fish and Wildlife Service is trying to figure out what the optimal recession rate is and water depths for the Everglades. This is telling us that birds have some flexibility and they adjust based on the standing stocks. And that was pretty cool finding for us. All right, this is just kind of a summary, but basically we're starting to fill in here now. We have some of the parameters that factors that produce high prey availability. We know something about the characteristics that birds use to select these patches. And then the next question is, well, is food really limiting? Because a lot of this assumes that that's the case. So we had that pretty interesting contrast I mentioned earlier, 2006 and 2007. Nest effort is the white bars. You can see there was a big drop in nest effort in 2007, about a 40% drop. And then the food that we were measuring in the dry season dropped by quite a bit more. So basically, there's a nice contrast. 2007 is the year I'll refer to as kind of the poor year. 2006 was a really good year. So is food limiting? I have a colleague, Mark Cook, at the Water Management District, who was out doing an experiment where he was taking white ibis nests, and half the nests he was giving about a 20% increase in biomass, and half the nests he would just handle measuring the chicks back. It was a cool trick. It worked. It was good. So Mark was a little bit concerned because after his first year, and many, many dollars and lots of effort, he found exactly no difference between this control and supplement. And this is bad news if you're doing an experiment, particularly if you ever want to publish it. But we were like, don't, remember 2006, we were in the marsh sampling food. We were just like, there's lots of food out there. Maybe it's the Everglades, maybe we should just wait a year and, we're bound to get something very different, right, in terms of hydrology. And sure enough, in 2007, it was a lot drier that year, and here's where we saw this big difference. So what this told us was absolutely, and this was basically survival, but we also looked at growth and pretty much saw the same trend. Can food be limiting birds? Absolutely it can. Does it every year? No, it doesn't. And that's also very good news for the restoration. All right, I was interested also in what's the effect of that limitation? What does it matter? We see it in reproduction, we see it in physiology. And so those same birds, same adults that we captured away from the nest, we took blood samples and did all kinds of work. Then we followed them to their nest, we sampled their chicks, looked at blood and a number of other measures. Basically, the bottom line was looking for measures of stress. We found that white ibises were more sensitive. They showed more stress during the year with poor habitat than did the gray egrets. Remember that cream skimmer crumb picker dichotomy I had? It's showing up again. I can go, I'm going to kind of skip this stuff, but we have some ideas about why that difference exists. But basically, we picked this difference up with measures of stress. We saw it in chicks. This is an example of nesting chicks. This is 2006, and again, the provision and control nest. So back to that experiment, we pulled blood on those birds as well. This is a stress measure called heat shock protein 60, or stress protein 60. We also looked at fecal cortical serum. It was the same pattern. Just like Mark Cook's data, no difference in 06, but in 07, significantly more stress in the control birds, suggesting that food limitation actually did have a penalty for the birds in terms of physiology. Are there any youngest siblings out here in the audience today? How many minutes do we have?
A few. Not many.
3, okay. Any youngest siblings? All right. You might be disturbed by this next slide. Not all the chicks pay the same penalty. Turns out if you're the oldest chick, that's the A chick in the bottoms, there was no difference in stress level between 06 and 07. Who pays the penalty? It's the younger chick. And actually the C chick, the third one, is not here, and there's a good reason for that. It's dead. So basically, and this is a pretty important mechanism actually for wetland birds. And you see this in predatory birds like raptors as well. That the adult at least has a chance to get off one chick in a poor year, and in a good year they'll get off the top of 2 or 3. But anyway, it was interesting because that's kind of goes along with that. All right, I'm going to skip through. It's only a couple more minutes. The last part I wanted to show you was sort of wrapping all of this stuff up. So we've got the map. We've hooked a couple of other studies to it, which now help us interpret our map data. Right? We now have a much better understanding mechanistically, and so we have to turn that information into some tool that the water management districts can use for the service or park service. And so we have both Joel and I believe kind of these simple models. This is a habitat suitability model. It works pretty well. I mean, we validate it with independent data. It gives the district a chance to sort of see what the landscape looks like from a bird's perspective. And it's a fairly simple model, but it incorporates lots of that other data that I showed you. Joel has a similar kind of approach for his fish data. He has a report card approach where he basically has targets based on historical data and historical hydro periods of what you should see out there in terms of fish communities. And there was his sampling sites, which are pretty extensive across the Everglades. And then he can score basically what the system is producing in a given year based on those targets. And it's a real simple report card approach, but it works pretty well for water managers. This is just particular, so you can adjust that basically based on particular species, based on the life histories in response to drought. And again, you can compile these kind of assessment kind of cards. So anyway, the bottom line here is we've got lots of information feeding into the map. If you can take that information and sort of package it in a way, a very simplistic way, it tends to be sometimes much more useful to managers, but it has a good scientific foundation. And we think that the map actually provides the ability to do that in the Everglades. And it might not be that common, but we should probably make use of it while we can.